Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. So, this is lecture number 27, where I will discuss several case studies on metabolic engineering applications in Catharanthus roseus hairy root system. So, the aim of course was to enhance the alkaloid content, and hairy root system was chosen. So, I will come to that. So, let me briefly tell the concepts what I am going to cover. So, first one is the expression of a MYB transcription factor gene MYC in hairy root culture. We will see what happened when attempts were made to express this transcription factor in hairy root. Next is the expression of ORCA3 and SGD gene in hairy root. The third one will be the expression of DAT gene in hairy root culture. The fourth one will be the expression of G10H either in singly or G10H ORCA3 together in hairy root culture. And finally, we will see the expression of DXS and G10H in one instance, in another instance DXS with anthranilate synthase ASA gene in hairy root culture. Let us now go to the board. So, this is metabolic engineering in Catharanthus roseus hairy root system, and of course, the aim basically was to enhance the alkaloid to enhance the contents of alkaloids. If I write that will be better, to enhance the alkaloids. Well, and the system used was hairy root culture. By now, it is clear to you that how genes can be inserted into hairy roots. So, basically this is what I used to call earlier, the same terminology I am going to use now. So, this is co-transformed hairy root cultures. So, if you remember that co-transformed hairy root cultures are raised by transferring without making any changes in the original RI plasmid, which is there for which is there in Agrobacterium rhizogenes, which contains the tDNA for RI, and of course, it has a virulence region. So, here recombinant plasmid has to be incorporated. So, which contains the synthetic tDNA construct having the border sequence and here you insert the gene or genes of interest. So, this is gene 1, this is gene 2. Of course, this will be under the control of promoter and terminator, which I am not showing here. And what should happen is basically this V will work both in cis as well as in trans. This work in trans, trans, and this is cis. So, it allows this tDNA to go out and this tDNA to go out. So, this is basically the tDNA of the synthetic plasmid. So, well, and then what will happen? So, both tDNA will integrate in the plant genome, and eventually this will be selected based on a marker which is an antibiotic selection marker. So, maybe a kanamycin resistance marker is there, and that leads to selection of, and so these two will integrate, and as a result of that what we are going to see is co-transformed hairy root culture, and this hairy root culture will express the genes like G1 and gene G2, and here it contains the ROL genes which is required for hairy root phenotype formation. Okay. So, this is in brief the mechanism of hairy root co-transform hairy root culture, and scientists whoever did this work, they prepared the synthetic plasmid which come under binary vector, and then they have inserted the genes there, and then they put the plasmid, recombinant plasmid inside the Agrobacterium rhizogenes, and then they did transformation in Catharanthus roseus young seedlings, and as a result of that hairy root emerges, and these hairy roots were checked using antibiotic selection pressure, and then finally they look for the growth performance, and then finally the product. So, with this let me first talk about case study 1, where expression of CRMYC1 gene in hairy root culture. I use this small terminology. So, that means MYC is basically MYC1 is a transcription factor, MYC1, MYC2 that plays important role. So, these are a positive regulator. So, the gene was put under the control of a promoter and a terminator, and then Catharanthus roseus co-transformed hairy root cultures were raised. So, we are not going to those details, we will see only in a nutshell what this co-transformed hairy root culture showed us, or what this hairy root culture produced. So, what we are going to see? We need to see that what happens to the content of the alkaloids. So, what they have decided? They have decided to measure several alkaloids, among which they particularly focused on azmalicin and catharanthine content. So, I do not think that I have to again mention about this. So, one route goes to azomylicin and other route goes to catharanthin. So, this formed by joining two substrate, one is secologanin and tryptamine. So, what happened, the outcome? The outcome is this, that azomylicin content enhanced by 13 to 14 fold as compared to the wild type hairy root. So, this is pretty enormous, and the catharanthin content also enhanced, but that is not that much, that is only 3 to 4 fold. So, to sum up, expression of Catharanthus roseus MYC1, which is a transcription factor gene, when this gene was transferred or overexpressed in hairy root culture of Catharanthus roseus, this leads to enhanced production of indole alkaloids, and particularly the impact was seen with the azomylicin content as well as in the catharanthin content. So, this is a metabolic engineering case study where positive result has come. So, next we go to the next case study. So, this is the heading, case study 2. So, here what we see, okay, one thing I forgot to tell, that this work was reported very recently in a journal called Horticulture, Environment and Biotechnology in the year of 2022, volume 63, page number 709 to 717. So, case study 2, we will see that okay, overexpression of ORCA3 and SGD in C. roseus hairy root culture. So, they have used the inducible promoter, which is called this glucocorticoid inducible promoter, though I will mention here promoter used. So, normally the promoter, what is used is cauliflower mosaic fibers 35S promoter. One is constitutive, example is cauliflower mosaic virus 35S promoter, but here this was not used. Here they have used glucocorticoid inducible promoter. So, when I will not go into this promoter details here, because otherwise we will not be able to finish the major outcome what we are going to discuss. So, here but of course, I will give you the reference and the details can be studied by the students themselves. So, here using this promoter, when they attempted to express ORCA3 alone, that did not make no significant changes happen in the total amount of alkaloid contents. However, when ORCA3 plus SGD, both genes were put in the construct under the control of a glucocorticoid inducing promoter, that leads to changes, detectable changes in the alkaloid content. So, significant changes detected. So, that includes increase in serpentine content, and by 44%, and azmalicin content by 32%, catharanthus content by 38%, and tabarsonin content by 40%. Not only that, it also showed increase in locnericin and horhamaricin. So, which I will write this side. And horhamaricin, this is by horhamaricin by 56% and locnericin by 60%. Such significant changes detected when both ORCA3 and SGD was expressed under the control of a glucocorticoid inducible promoter. Now, this is very interesting because this is root, and what I have said yesterday, that if we briefly look into the pathway, that is this tabarsonin in root. Tabarsonin is not converted into vindolin. Instead, different pathway operates in roots that leads to the formation of locnericin and horhamaricin. So, what has been found that tabarsonin content enhanced, catharanthin content also enhanced, and the pathway leads to assimilation content enhanced. Not only that, serpentine content also enhanced. So, this is a very positive result, and this paper published in a journal called Protoplasma in the year 2016. Volume 253, page number 1255 to 1264, and there are two authors. So, one is Sun and Peebles, and they are from Colorado State University, USA. So, this is another positive result with metabolic engineering application. Let us go to the next case study. Next case study, let me go to the first slide, DAT genes. So, case study 3. So, this is expression of diacetyl vindolin 4-acetyl transferase, that means expression of DAT gene in roseus hairy root culture. So, here what they found. So, the same co-transform hairy root they have used system, and the DAT gene was expressed. So, DAT, you know that this is the more or less the terminal step leading to the formation of vindolin. So, if I put the pathway, so that means I think that is required to explain. Saying this way, if I write tabarsonin, tabarsonin 1, 2, 3, 4, deoxyvindolin, NMT, this is D4H. Now T16H, then is T16 OMT, third one is T3O T3R, then comes to NMT, and then D4H, and then last one is the DAT, which makes vindolin. And tabarsonin in the root system, it produces lock rocknericin, or from there it can produce or hemaricin. Okay. Now, what happened as a result of that? So, the first of all, how they have studied? They have studied they have checked the enzyme activity of DAT enzyme activity they have checked, and next what they have checked? They have checked the metabolite content. So, what they found? DAT expression leads to the accumulation of horhamycin. So, what happens when DAT expressed? That leads to accumulation of horhamycin. That is pretty interesting. Where is horhamycin? It is here. Why it is so? Because DAT is basically situated here in the pathway, okay. But what is also known to some extent, that there are enzyme like T67E, like MAT, and then there are enzyme like T19H. So, here so the MAT, and this is T67E. T67E. Now, this MAT, the full form of MAT is minovenecin 19O acetyl transferase. So, that means this MAT and DAT is more or less similar type of enzyme. So, when you overexpress DAT in the hairy root of Catharanthus, what happens? That DAT which is meant for vindolin, it is not working, because in order to have vindolin accumulation, you need to have the light. So, that is not happening, but the DAT enzyme is produced in higher amount, but what happens in the dark, the pathway from tabarsonin moves in different direction like locrenin and horhamycin. So, this is operating in the root, constitutively operated. So, the extra copy of DAT is rather now working together with MAT, and that leads to the increase of flow in this direction and leading to more or hemaricin accumulation. So, this is an interesting paper. So, this is what is the explanation I try to provide. So, I just put reference phytochemistry 2007, volume number 68, page number 1922 to 1931. So, this is a paper from Canada led by Vincenzo Di Luca. So, the next we go to the case study 4, where I will use here only case study 4. So, where basically the overexpression of G10H and ORCA3 leading to improved catharanthin production. I already told you the outcome. Okay. So, G10H is one of the early enzyme I have not covered in that details. So, which basically converts the geraniol to 10 hydroxygeraniol, and this subsequently takes part in the formation of secologanin. Okay. And ORCA3 we know that is basically a transcription factor. It affect the pathway. So, what happens that when they have used only G10H expression, that also leads to changes in alkaloid levels, but when both G10H and ORCA3 were expressed, that made subsequent changes and produced more catharanthin, which is very prominent, more catharanthin compared to the other alkaloid, more catharanthin was detected. And interesting point is this, in this hairy root line, when they fed abscisic acid, that means feeding of abscisic acid in co-expressed a hairy root, that leads to the catharanthin accumulation up to the level of 1.96, which is almost 2 milligram per gram dry mass of hairy root. This is also very interesting outcome. and this result published in the journal called Plant Cell Reports in the year of 2010, volume 29, page number 887 to 894. And the last case study which I will cover very briefly, so which is case study, this will be case study 5. Where DXS plus G10H in one instance, and another instance what they have used DXS plus anthranilate synthase. So, this they have said ASA. So, DXS is the pathway which is basically the first enzyme of the MAP pathway, and G10H is basically it is towards the end enzyme of the MAP pathway. That means, when you are expressing both DXS and G10H, that leads to more secologanin formation, and similarly in other case use DXS. So, DXS contribute to this is the first enzyme of the MAP pathway that leads to formation of secologanin eventually, whereas the ASA, which is anthranilate, which is product of the Shikimate phenylpropanoid pathway, and eventually this leads to the formation of more tryptophan. So, that means more tryptamine, and more tryptamine, more secologanin, that leads to the formation of strictocidin, and here this secologanin joins with the normal tryptamine level and makes strictocidin. So, let me briefly tell what is the outcome of this. So, outcome in general that it increases the indole alkaloid accumulation. So, DXS overexpression resulted in significant increase in azmalicin, serpentin and locnericin. So, azmalicin, I use a different now. Serpentin and what I say the locnericin, that all enhanced not by locnericin by 49% upliftment, azmalicin by 67% upliftment, okay, and serpentin by 26% upliftment. This happened, and when both DXS and G10H was used, so that leads to cause significant increase in azmalicin, locnericin and tabardassonin. So, I am not showing the data. Likewise, when DXS and ASA overexpress, that displayed increase of horhamaricin, locnericin locnericin and also tabarsonin. So, that means tabarsonin content enhanced or hemaricin content enhanced as well as locnericin content enhanced. So, here some data was provided, tabarsonin enhanced by 34%, horhamycin enhanced by 30% and locnericin enhanced by 27%. That means, this is also very positive outcome as a result of that, because you know that it is basically enhancing the strictocidin and that eventually leads to formation of all downstream products. So, this paper published in the journal called Metabolic Engineering in the year of 2011, volume number 13, page number 234 to 240. It's from United States, the same group. So, these are all the case studies which I have discussed in this class, all showed positive impact on the levels of indole alkaloid accumulation in Catharanthus roseus. So, this is what is the take home message, and with this I end this class. Thank you.